species richness in open habitats compared to those in the interior of forest or on the edge of the forest. The environmental conditions vary in temperature, moisture, sunlight, and airflow, which provides niches for many different organisms, with the edge facilitating the movement of matter between them. With this context of different habitats, we sought to understand the role and effect of these conditions on fungi, as all fungi, regardless of classification, require oxygen, water, carbon, nitrogen, and multiple different micronutrients to grow. These requirements seem to be present in all of the habitats at differing levels. Fungi are essential to the environments they find themselves in and produce carbon dioxide, water, and many other byproducts that affect and facilitate the growth of other organisms.
The goal for collecting this data was to better understand the ecological role fungi play at a forest habitat's edge. Our original hypothesis was that the species richness of fungi is higher along the edge of a forest compared to the interior or the clearing. After encountering issues along our data collection journey, more specifically the lack of visible fruiting bodies in the soil due to freezing temperatures, our focus shifted to fungi colonies growing on downed trees along the edge, interior, and in clearings. As we set out to research, we visited Hone Quarry, Hillendale Park, and Cooks Creek Arboretum for data collection. Using a soil master for moisture level and pH, a densiometer for canopy coverage, and a caliper for measuring the size of individual fruiting bodies.
After having to adjust our project according to the limitations of both field studies and our equipment, we gathered different data points in order to look for a trend in the data, hoping that one of them might have a correlation. Our main data point was going to be the average size of the individual in hopes that it might be conducive to the conditions offered by the different habitats. While that didn't prove to have any correlation we could observe, we also measured the pH and moisture levels at the different sites of the organisms found. We found that neither pH nor moisture levels were exhibited at significantly different amounts between the habitats.
As we mentioned before, we found many limiting factors along the way that caused us to amend our concentration. Visible fruiting bodies were lacking because of the drop in temperature as our data collection progressed. Rain levels differed and skewed our data since our data acquisition occurred over a few weeks rather than one single day where the moisture levels would have been more standardized. The effectiveness of scientific instruments was called into question as well, and we're not sure just how accurate the information gathered from our soil meter really was. And lastly, conducting a small-scale research study presents challenges as we only gather data on 30 down trees with visible fungi colonies, whereas on a larger scale, significant differences could have been found. While researching, we were not able to find similar studies testing edge effects on fungi, so we hope to open the door to this experiment on a larger scale in order to understand fungi species' ecological interactions with downed trees and how the edge facilitates these interactions.